Hello students, in continuation of my earlier module on soil formation and structure, I'll be discussing in this module about the macronutrients and micronutrients found in the soil, what is their main role in the soil, how does the ions exchange takes place in the soil, how does it influence the fertility and productivity of the soil, as well as what is the nitrogen cycle will be discussed in detail in this module. Our learning objectives in this module will be about macronutrients, micronutrients found in plants and their functions, the soil ion exchange reactions, how does pH of the soil influences the productivity and fertility of the soil, and the nitrogen cycle which takes place in soil. So the first question which comes into our mind is, what is fertile soil? The soil that produces abundant crops under the appropriate environmental conditions is termed fertile. The soil fertility reflects the soil-plant relationship with reference to plant nutrients available in the soil. Understanding the principle of soil fertility is important because it leads to efficient nutrient management. It is profitable in terms of crop protection, and it also helps in environmental protection. If you compare productivity with fertility, then soil fertility is the innate capacity of soil to provide adequate amount of nutrients essential for plant growth during favorable conditions such as temperature, water and light, and the physical conditions of soil. Whereas soil productivity is the economic concept that signifies the capability of soil to produce specified plants under well-defined and specified systems of management inputs and environmental conditions. It is a function of several factors rather than being a soil property alone. Remember, not every element can be regarded as essential. For an element to be called as essential, it needs to meet the following criteria. The plant cannot complete its life cycle without it. That is, completing the life cycle means it grows from seed to new seed. If the element is specific in its physiological function in plants, then also that element will be regarded as essential. If the melody that develops in plants can be remedied only by that element, then it will be termed essential element. Also, the element function cannot be replaced by any other element, and that element is directly involved in the plant growth and reproduction. So, looking into these features, 18 elements have been identified as essential for the growth of all plants. They are further classified not on their relative importance, but on the basis of their relative amounts into macronutrients and micronutrients. Macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, potassium, calcium, magnesium and sulfur, whereas micronutrients include boron, copper, iron, manganese, zinc, molybdenum, chloride, cobalt and nickel. Now coming to the macronutrients, as already been told to you, these elements occur in ample levels in plant biomass and fluids. That is why they are known as macronutrients. They are further divided into three classes. First is the structural nutrients, which includes carbon, hydrogen and oxygen. They are fundamentals for carbohydrates and they serve as energy sources. Then we have primary nutrients, nitrogen, phosphorus and potassium. They control functions of plant enzymes and contributes to plant nutrient content. Then we have secondary nutrients, calcium, magnesium and sulfur are the examples of it. They control soil acidity and are involved in various plant processes. As far as their uptake form and plant mobility is concerned, carbon is taken in the form of carbon dioxide and bicarbonates, hydrogen as hydrogen ion, hydroxyl ion, oxygen as oxygen molecule, nitrogen as nitrate and ammonia molecule, and their mobility in plants is shown here in the table. They are mobile in plants and mobile as nitrate and immobile as ammonium ion in soil. Phosphorus as dihydrogen phosphate, which is somewhat mobile in plants and immobile in soil. Potassium is very mobile in plants and somewhat mobile in soil. Calcium has calcium ion, which is immobile in plant and somewhat mobile in soil. Magnesium has magnesium ion, which is somewhat mobile in plant and immobile in soil. And sulfur has sulfate, which are mobile in plant as well as in soil. As far as their functions are concerned, carbon, hydrogen, oxygen, which are the foremost components of carbohydrate, proteins and fats, they are almost 94% of dry weight of plants. They aid in oxidative breakdown of carbohydrates, proteins and fats during cellular respiration. They provide energy for the growth and development of plants. Nitrogen has a key role in respiration and synthesis of proteins. They maintain fertility when bound to humus. The increased tillering and promotes vegetative growth of food crops. Their deficiency decreases in rate and degree of protein synthesis as well as development of chlorosis. On the other hand, phosphorus, which makes up for structural component of the membrane system, it has a resistance to diseases in plants. It also influences cell division, root growth, fruit and seed development. The deficiency of phosphorus is restricted to root and shoot growth. It also leads to shedding of leaves and delaying in flowering. The potassium, which is responsible for maintenance of water balance, is indispensable constituent of chlorophyll. It protects against fungal and bacterial diseases, and its deficiency leads to development of chlorosis and drying back tips of shoots and reduced growths of shoots. Calcium, which is responsible for control of soil acidity, is also involved in root elongation and cell division. It acts as a buffer in plant system. It improves intake by correcting the pH of soil, which will be discussed further in detail, and its deficiency will lead to wrinkled leaves, shorter roots, brown growth of plants. Magnesium, which is the main constituent of chlorophyll, controls uptake of nitrogen and phosphorus from soil. It acts as a catalyst in activation of enzymes, and its deficiency leads to premature defoliation. Sulfur, which is another macronutrient, promotes module formation, root growth and seed formation. It increases oil content in oil-bearing plants. It is essential nutrient for cereals and sugars. Its deficiency will lead to brittle leaves and stems in plants. Now coming to the micronutrients, as already told to you earlier, these nutrients are required in lesser amounts and as they are toxic at higher levels. They stimulate plant growth and actively participate in photosynthesis. They show catalytic behavior in plant processes. Their deficiency hampers plant growth, photosynthetic rate, and that reduces crop yield. Their deficiency may cause shedding of flowers, poor seed settings, and attack of disease. The microelemental nutrient, their uptake form and plant mobility is shown in this table, where typical range of soil content is also given. This is being mentioned in milligram per kg except where percentage is used. For example, we have iron, which is taken in the form of ferrous and ferric. They are immobile in plant and soil and their range is 0.01 to 2.1%. Boron is taken as borate and boric acid. They are immobile and very mobile in plant and soil respectively, and their range varies from 0.9 to 1000 milligram per kg. Similarly, copper is immobile in plant and soil and it ranges from 2.5 to 60 milligram per kg. Chlorine is mobile in plant and soil. Its range is 18 to 806 milligram per kg. Manganese is immobile in plant but mobile in soil, and it varies from 1 to 18,300 milligram per kg. Molybdenum is taken as molybdenum oxides. They are immobile and somewhat mobile in soil, and their range varies from 0.07 to 5 milligram per kg. Zinc is immobile in plant as well as in soil, and the typical range is 1.5 to 2000 milligram per kg. Cobalt is immobile in plant and somewhat immobile in soil, and it varies from 0.3 to 200 milligram per kg. Nickel is mobile and somewhat mobile in soil, and its range is 0.1 to 1523 milligram per kg. As far as their functions are concerned, iron aids in chlorophyll formation, whereas boron regulates nutrients, production of sugar and carbohydrates, and is also essential for fruit and seed. The copper, which is important for proper reproductive growth, it helps or aids in root metabolism and helps in the consumption of proteins. Manganese, which functions with enzyme systems, it is involved in breakdown of carbohydrates and nitrogen during metabolism. It regulates the intake and state of oxidation of various elements. Chloride, it helps in carbohydrate metabolism in the plants. It stimulates enzymatic activity. Whereas molybdenum helps in the use of nitrogen and in nitrogen fixation, as it is a constituent of nitrate reductase and nitrogenous enzymes. Zinc, it contributes in the mechanism for the transformation of carbohydrates and consumption of sugars. It also constitutes a part of enzyme systems which regulates the plant growth. On the other hand, cobalt, which plays an indispensable role in vitamin B12 production, which assists in the process of nitrogen fixation also. It affects plant growths as well as metabolism. Nickel prevents urea accumulation to toxic levels. It prevents leaflet tip necrosis. It is essential for the mobilization of stored seed nitrogen. Now coming to the cation exchange capacity, which refers to the capacity of the soil to exchange cations to enhance availability of essential trace metals to plants. Mathematically, it is defined as the number of milliequivalent of monovalent cations that can be exchanged per 100 gram of soil. The value of cation exchange capacity, which is commonly called as CEC, varies with soil conditions and pH. CEC increases with presence of negatively charged sites on minerals. Remember, humus has high cation exchange capacity, which is almost 300 to 400 milliequivalent per 100 grams, while that of organic matter soil is 10 to 30 milliequivalent per 100 grams. The anion exchange capacity is the measurement of the positive charges in soils being affected by the amount of negative charges which a soil can absorb. The value of AEC is directly proportional to change in pH, while in case of cation exchange capacity, reverse trend is seen. The relative strength of adsorption of cations and anions can be illustrated by lyotrophilic series, which is shown here. For cations, this capacity is maximum for aluminium, then hydrogen ion, calcium, magnesium, potassium, ammonium and sodium ion. But for anions, it is maximum for dihydrogen phosphate, sulfate, nitrate and then chloride. As far as exchangeable sodium percentage is concerned, this property reflects the alkalinity of soil too. If it is greater than 15, then soil gets dispersed, causing sodium toxicity. It becomes impervious, which means the water cannot pass through it and hence unfavorable for plant growth. The availability of plant nutrients is reduced and crop growth is affected. The exchangeable sodium reacts with carbon dioxide, forming sodium carbonate, which on accumulation renders soil alkaline and ultimately it is infertile. You can see from these equations, soil sodium, which is there attached to soil, is converted into sodium carbonate with the reaction of carbon dioxide. So soil ion exchange has lot of significance in soil fertility. The base saturation of soil is an indicator of the degree of weathering. cation exchange capacity supports the clay content, which is useful for soil management purposes. CEC is an indicator of the nutrient storage capacity of soil also, and the information of this exchangeable cations is used in soil classification too. Now coming to the soil pH, which is also an important parameter of soil fertility, and in fact soil pHs is the measure of soil acidity or alkalinity. It is a master variable. It affects chemical, physical and biological properties of soil. The optimum pH for most crop should be between 5.5 to 7 because it gives you the maximum nutrient availability. Metal toxicity and solubility is also determined by the pH. For example, aluminium toxicity is seen in the soils which have pH less than 5.5. The microbial activity is also influenced by pH, especially important in the nitrogen cycle. The pH having 6 to 8 is seen to have the highest nitrogen content. pH which have 6 to 7.5 is high in phosphorus content. pH less than 6, which is known as acidic soils, it has formation of insoluble compounds of phosphorus with iron and aluminium, and pH having greater than 6 in soils which are being classified as alkaline soils. Here the formation of insoluble compounds of phosphorus with calcium is commonly found. Now these pH can be also changed depending upon whether the soil is basic or acidic. If you have basic soils, then their pH can be reduced by addition of iron or aluminium sulfates as well as sulfur in forms of sulfuric acid. Even addition of urea, ammonium, their nitrate, sulfate and phosphate forms, as well as monopotassium phosphate fertilizers are used to reduce the pH. Even addition of pine needles, sawdust and acid peat will also reduce pH. The decomposition of organic matter in the form of plant litter, compost and manure will also decrease the pH of basic soils. On the other hand, the pH of acidic soil can be increased by addition of lime like calcium carbonate or magnesium carbonate, by addition of wood and oyster shells, even by adding calcium silicate which removes free hydrogen ions, which will in turn leads to neutralization of active acidity, will change acidic soils to basic soils. Remember, pH value near neutrality is the best suitable for growth of plants. Here I have shown you some examples. If the soil becomes too acidic, it can be refurbished by liming. You can see it from here. The hydrogen ion is replaced by calcium ion and you have soil which becomes greater in pH and moves towards basicity. If the soils becomes too alkaline, especially in areas receiving scanty rainfall, it can be treated with aluminium or iron sulfate, which releases acid on hydrolysis and will change it into acidic soils. Sulfur is added to soils is also oxidized bacterially mediated by certain reactions to sulfuric acid, which will also lead basic soil to move towards acidity. Now here is the nitrogen cycle, where molecular nitrogen from air is fixed by soil organisms. The nitrogen enters into the bodies of soil organisms and organic substance, which is commonly humus in soil, through the process of decomposition and is further recycled through plants and animals. Now I'll be showing you the video which gives you an importance of nitrogen cycle as well as how does this nitrogen cycle takes place is in the soil is explained beautifully.
This section of the video is about the nitrogen cycle. Nitrogen is important for life. Proteins are made from it, and if you're talking about the use of nitrogen, it's for protein, which is for growth. The first thing you need to understand is the forms of nitrogen. that you might find in the nitrogen cycle. So you've got nitrogen gas, N2, you've got nitrates, which is an iron, NO3-, and you've got ammonium ions, NH4+. Now you might also hear of nitrites, which are related to nitrates, and ammonia itself, which mixed with water makes the ammonium ion. So the nitrates are the form in which nitrogen is absorbed by plants, and the ammonium is mainly associated with decaying material and waste products. Once you understand the forms of nitrogen you can have, this next stage is to understand what the bacteria do, the role of the bacteria in the cycle. So there are 3 types of bacteria, and they do one of 2 things. They either make nitrates or they remove nitrates. Denitrifying bacteria remove nitrates, denitrifying, remove nitrates. Nitrifying make nitrates from ammonia or ammonium compounds, and then we've also got nitrogen fixing bacteria which make nitrates from nitrogen gas directly. Now here is a diagram of a nitrogen cycle. Can you fill in the gaps? Right, first of all let's check what we've got. We've got plants and animals, and then we've got nitrogen in the air spaces in the soil, as nitrates in the soil, and as ammonium ions, and then these 4 boxes here are where our bacteria go. So first of all, can we explain what the arrows do? Well, we've got nitrogen in the air up here and nitrogen in the air down here, so that's just movement of air, so we can do that one. Movement of air or diffusion of air into the soil. Then how does the nitrogen as nitrates get into the plants? Well, that's just going to be absorption by the roots, so nitrates are absorbed by the roots. Then plants, nitrogen in plants getting into animals is going to be by eating, and then from animals into the soil is going to be by death and decay. So that's fairly straightforward, I think. The tricky bit always will be getting the bacteria in the right places. So let's see what we remember. Denitrifying bacteria remove nitrates. We've got an arrow here which is going from nitrates in the soil to nitrogen in the air. So this removal of nitrates in the soil is going to be denitrifying bacteria. Then we've got making of nitrates, and we've got making of nitrates here from ammonium ions, ammonia containing compounds, so that is going to be the nitrifying bacteria. And then finally we've got the nitrates being made here by bacteria directly from nitrogen in the air spaces, and here we've got nitrogen being incorporated into plants by bacteria. So they are both nitrogen fixing bacteria. Now I just need to explain this part a little bit, and the term legumes. Legumes is a term given to certain type of plant which have root nodules. So they're little nodules on the roots that actually contain nitrogen fixing bacteria. So they are kept in there by the plant to directly convert nitrogen gas into nitrates for the plant. So examples are beans, peas and clover. So they're very good for getting nitrogen from the air to the plant. And then finally, the thing we haven't mentioned yet is lightning. A lightning strike has sufficient energy to combine nitrogen and oxygen directly into nitrates.
We come across many terms in this nitrogen cycle which was shown to you in the YouTube video. The terms associated with this ammonification. This is a process in which nitrogen containing organic molecules present in the soil is converted into ammonium compounds. The amount of accumulation of where ammonium varies with organisms, soil and environment. Then we have nitrification, which is a process in which ammonium or other reduced forms of nitrogen are oxidized to nitrite and nitrate. With the help of nitrifying agents, which includes nitrosomonas and nitrobacter, the nitrate formed is utilized by crops and soil organisms for nutrition purposes. The third term, which is commonly used in nitrogen cycle, is denitrification. This is a process of conversion of nitrite and nitrate into molecular nitrogen. This is caused by Pseudomonas and Bacillus and Parococcus. This is an anaerobic condition of soil which is conducive to denitrification. As far as significance of this nitrogen cycle is concerned, this leads to the formation of microbial cells and organic matter in soil and its intermediate byproducts, which leads to the formation of humus in soil. So students, these are further suggested reading material for this module, which you can go through it. And in this module, we have learned about the various nutrients which can be classified into macronutrients and micronutrients, and about the role it plays in soil productivity and fertility, and how and in which form they are available to the soil, and also how does soil pH influence the fertility and productivity of the soil. We have also seen the common nitrogen cycle which is taking place in the soil, which leads to ultimate good soil formation. Thank you very much.